Hi, this is Mr. Cho, and today I'll be teaching you how to use the O-level syllabus document. Okay, so this is easily available online. You just need to go Google and search for O-level science syllabus, and you'll find this. Okay, now very first thing is that please be aware that this year 2021 there will be common last topic removal, and so you can look through these documents and you can see which are the exact questions or exact learning outcomes that have been removed. For bio, of course, that includes the last chapter on ecology. For your chemistry, right, for those who are taking 5078, you will realize that there's one removal of the CLT which will result in you having no question option in section B of your chemistry paper. And some details you can read for yourself. The duration is unchanged and there is fair assessment. Alright, now, so why did I show you this document? All of you should have this document. If you do not have it, you need to print it for yourself. Now in this document, you can find all the learning outcomes and learning objectives for each chapter for all your sciences, right? Physics, chem, and bio. Now today I'm talking about bio, of course. And so why is this document important for you to look through? It's basically a checklist for you to see if you have understood everything from the chapter. Okay, so let's take for example chapter 1, right? Cell structure and organization. So you can see that the very first learning outcome is that candidates you should be able to identify cell structures, right? The organelles of the typical plant and animal cells from diagrams, photomicrographs, which means this thing is called electron microscopy, as well as light microscopes. And you need to identify these organelles: chloroplast, cell membrane, cell wall, cytoplasm, cell vacuoles, nucleus, mitochondria, ribosomes. Okay, so you see that there's 6 of them here. So these 6 has to be observed under photomicrographs and light microscope. But there's 2 mitochondria and ribosomes that can only be seen under electron micrographs. Now let's look at some examples online. So this is what I mean by electron micrographs. Now let's look at these 2 features. Right, so this is a electron micrograph of an animal cell. Now the very first thing you should be able to identify is the nucleus, right? So where's the nucleus? It'll be over here. So for the plant cell, you can see that the nucleus is over here. Now usually you'll be able to see a dark black dot in the nucleus. Sometimes in the animal cell you'll see patches here and there, but it's really about looking for that black patch. Now we didn't learn about this ER, but we learned about mitochondria, right? So mitochondria in the animal cell, right, it looks like a little worm and sort of see lines going in between. In a plant cell, you will see that the mitochondria is usually much smaller than the chloroplast. Right, so here's a chloroplast. In the chloroplast, you might be able to see certain structures in the chloroplast that these are the chlorophyll molecules. Of course, in the plant cell, you have a cell wall. Right, so there's a difference, right? We see 2 differences already. The plant cell has a cell wall, and the plant cell has chloroplast, which the animal cell does not. The third difference, of course, would be the vacuole. In the animal cell, there are numerous small vacuoles, whereas in the plant cell, there's one large central vacuole. Now, what do these organelles do? Let's do a quick recap. Right? So if you look at C, right, you need to state the function of the organelles identified above, which means that you need to be able to know all of these organelles. Let's take a look at your textbook. Right, so you can see starting from the cytoplasm, right? The cytoplasm is the site where most cell activities will occur, and this is where all the organelles are contained inside. Then we talk about nucleus. So nucleus here, we know that the nucleus, it controls the cell activities such as cell growth and the repair of worn out parts, and it is essential for cell division. So cells without a nucleus, for example the red blood cell, will be unable to divide, be unable to repair its worn out parts, and will generally die faster than a cell which has a nucleus. So what you're going to do is to look through each of these organelles and see that you know the function for each. Now next, let's see. Compare the structure of the typical animal and plant cells. Now we have just done that with our micrographs here, right? But just to drive the point in, let's just go Google and search for light microscope of animal versus plant cell. And we can see, let's see this picture. Right, so in this picture you can see the plant cell. Right, you can see the cell wall, you can see the chloroplast, and if you look really carefully, right, at this little dot there, that is the nucleus. Whereas in the animal cell, in the light microscope, you can only see the nucleus. These small dots are only if you have a very special stain. Okay, so normally you should only see the nucleus, cytoplasm, and the cell membrane. That's all. Can't even see the ribosomes. Alright, so then let's talk about E. Alright, stating in simple terms the relationship between cell function and cell structure in the following. Right, absorption in the root hair cells, conduction and support in the xylem vessels, and transport of oxygen in the red blood cell. So what does this mean? What does it mean to state the relationship between cell function and cell structure? For that, we need to look at our textbook again. So we find the relevant information. Now in the textbook, the thing is that there's a lot of information that might not be as important or as relevant as what is written in the syllabus document. So the syllabus document really is meant to guide you to the more important parts. Okay, that's not to say that the whole textbook is useless. Textbook is very important, right? You just need to be able to identify which parts are more important than others, because your time is limited. So this one here, right? How is the cell structure related to the cell function? So here we see the structure. Right, the structure of the red blood cell pretty simple, with a biconcave shape. Right, the cell is a biconcave shape, and it contains hemoglobin, and it does not contain a nucleus. So that's the structure. And what are the adaptations for the function? This requires you to write a bit more. So we see the red blood cell contains hemoglobin, which binds to oxygen and transports oxygen around the body. The red blood cell has a circular biconcave shape, which will increase the surface area to volume ratio, and this allows it to take in and release oxygen at a faster rate. It also lacks a nucleus, and this enables the cell to store more hemoglobin, which can bind to more oxygen, so it can transport more oxygen. Same thing for xylem vessel, right? You got to talk about the structure. So you see the structure, there's a lumen. A lumen is an empty space, and basically that's it, right? It's actually these are dead cells. So the xylem vessels are long hollow tubes extending from the roots to the leaves, and they are narrow and have no cross walls to obstruct water flow through the lumen. They do not have protoplasm to offer resistance to water flow. Their cells are thickened with lignin to prevent the collapse of the vessel. Right, so pretty much just some memorization stuff. Last one, root hair cell. You see that there's a long and narrow root hair, and there's a nucleus. Right, no chloroplast here. Why? Because the root hair cell is underground. It does not see, there's no sunlight underground, so there's no need for chloroplast here. So the root hair cell has a long and narrow root hair, and this increases the surface area to volume ratio of the cell. Right, water and mineral salts can be absorbed at a faster rate. Now do take note, water will enter in the root hair cell mainly through the process of osmosis, whereas mineral salts are actually diffusing into the root hair. So in those examples that I just given to you, you saw how I went to a textbook to find for information to answer this question. Now that doesn't mean that just by reading a textbook alone, I can be sufficiently prepared for all the questions, right? I need to use the knowledge gained in this section in new situations or to solve related problems. That means that once in a while, you're going to get questions that test your ability to think creatively, but you still need to refer the, to base your answers from your knowledge in the textbook. Okay, so you're not really going to write very random things. You should be writing things that you have learned before. Okay, maybe just in a different or more creative structure. Alright, now what I want you to do is to go through each chapter, right? Each chapter and look through each learning outcome. Ask yourself, are you able to do each of these learning outcomes? Are you able to define diffusion? Are you able to describe the role of diffusion in nutrient uptake and in gaseous exchange in plants and humans? Are you able to define osmosis and describe the effect of osmosis on plant and animal tissues? Right? For example, if you have a red blood cell in a solution with higher water potential than the red blood cell, what's going to happen to the red blood cell? Right? Is it going to burst? Is it going to lyse? Or is it going to shrink and crenate? If you're not sure what I just said, then you need to go and read up. Okay, so all of this, go through one by one. And if you can, get a pencil or some pen and just check it off, right? As you do, check it off, and make sure that you can do all of the tasks here. Okay, so this is the basis of how you can do your revision. I'm showing you this document to guide you on how to use it. If you do not use this, you'll feel very lost. You don't really know where to study in your textbook. As I just said, textbook is pretty big. There's a lot of extra information that might not be so important as for the test right now. Okay, of course it'll be useful just for general knowledge. Okay, so I hope this guide helps you, and I wish you all the best for your prelims. I'll probably see you around doing your tests. Okay, alright, all the best for your slides. Bye. That's all.